For over a century, Pompeii's plaster casts told a clear story: mothers shielding children, sisters locked in a final embrace. Then scientists extracted ancient DNA from bones still sealed inside those casts. The two maidens, both male, the mother cradling her child, a man holding a boy he had never met. Nearly every identity was wrong, and understanding how they were misidentified begins with a technique invented in the 1860s. When excavators uncovered the entwined figures in the house of the Cryptoporticus in 1914, director Vittorio Spinazzola described them as le due fanciulle, the two maidens. His notes lingered on the softness of their limbs, the gentle arc of their embrace, and the supposed delicacy of their posture. For decades, museum labels and guidebooks repeated his words, painting a picture of tragic sisterhood, two adolescent girls, perhaps siblings, clinging to each other against the onrush of ash. Photographs and postcards cast them as icons of feminine vulnerability, a tableau of innocence lost to disaster. Yet beneath the plaster, the reality waited. In 2017, a team led by Estelle Laser used high-resolution CT scans to peer inside the casts. The pelvic shapes told a different story. Both skeletons showed the robust features typical of males: broad sacra, pronounced muscle attachments, and, in the younger figure, a growth plate consistent with an 18-year-old boy. The older individual, once thought to be a protective mother or older sister, also matched a male profile. The scan results, published in the Journal of Archaeological Science, challenged a century of visual assumptions. Could the bones have been misidentified or altered during early restoration? The final verdict came with DNA. In November 2024, researchers from Florence, Harvard, and the Max Planck Institute extracted ancient DNA from bone fragments sealed inside the casts. Genome-wide analysis confirmed both individuals were genetically male. No maternal or paternal kinship linked them. No sisters, no mother and child, not even distant cousins. The two maidens were two unrelated young men, their true identities hidden for generations by the seductive logic of posture, clothing, and cultural expectation. The myth, so carefully built on outward appearance, collapsed under the weight of molecular proof. Beneath the collapsed staircase of the House of the Golden Bracelet, a tableau seemed to confirm every expectation of Roman family tragedy. The central figure, draped in a flowing garment, wore a heavy gold bracelet. Over 1/2 kilogram of twisted filigree, unmistakably expensive and, to 19th century eyes, unmistakably feminine. Curators and guidebooks for generations described a mother shielding her child, a scene of maternal courage and loss. The child, nestled close, was said to be 5 or 6 years old. Two more adults nearby were cast as male protectors, a father perhaps, or brothers, rounding out the image of a nuclear family frozen in its last embrace. Yet, this story too was built on posture and ornament. The bracelet's weight and design, long read as evidence of a woman's status, became a clue that demanded further scrutiny. Metallurgists from the museum's conservation lab cataloged the piece, a robust copper-silver alloy, not the delicate goldwork typical of elite Roman matrons, and not restricted to female wearers. The narrative began to wobble. The real break came in November 2024, when genome-wide DNA analysis was performed on bone fragments from all 4 individuals. The results were unequivocal. Every person in the group was genetically male. The supposed mother was a man, the child a boy of about 5, and the 2 other adults also unrelated males. No kinship linked any of them, not even as distant cousins. The group, once imagined as a family, was in fact a gathering of strangers or acquaintances caught together in a moment of chaos. The gold bracelet, rather than a marker of motherhood, was simply a possession, perhaps grabbed in haste or worn as a symbol of status by its male owner. The old tableau, so carefully constructed from surface impressions, had dissolved under the pressure of molecular evidence. Inside the conservation labs of Florence and Naples, a new kind of investigation began. The plaster casts, so long treated as finished artifacts, became subjects for forensic scrutiny. 14 casts were selected, each with a tangled history of restoration. Some reinforced with iron rods, others patched with modern plaster, their original bones sometimes jumbled or missing. The challenge was immense. Ancient DNA, already fragile under the best conditions, was now sealed inside a mix of gypsum, animal glue, and volcanic ash. Any modern contamination, skin flakes, dust, even the sweat of a gloved hand, could swamp the faint signal of the past. Technicians used rotary tools to shave away the outermost layer of bone, then blasted the fragments with ultraviolet light for 45 minutes to destroy surface DNA. Only after this decontamination did they grind the bone to powder, extracting what little genetic material remained. The team adopted a partial UDG protocol, a careful chemical treatment that preserves the unique damage patterns of ancient DNA, those telltale cytosine to thymine changes at the ends of molecules, while reducing the risk of false positives from modern sources. Every sample was barcoded and double-indexed before sequencing, then compared against a 1.24 million SNP panel used in studies of ancient Rome and the Mediterranean. Out of 14 attempts, only 5 yielded enough human DNA to reconstruct a genome, but these 5, painstakingly authenticated by damage profiles and contamination checks, became the foundation for everything that followed. The results, published in November 2024, set a new standard for ancient DNA work in extreme environments. The same protocols that had succeeded in permafrost and desert graves now proved their worth in the hot, mineral-rich tombs of Vesuvius. With these genomes in hand, the team could finally test the stories that had been built on posture, jewelry, and imagination alone. Genetic data from the Pompeii casts not only rewrote individual life stories, it redrew the city's entire social map. When researchers charted the genomes of 5 victims against a reference panel of over 1,000 ancient Mediterranean individuals, a pattern appeared that defied every 19th century assumption. Instead of clustering with ancient Italians, the Pompeians grouped tightly with Eastern Mediterranean populations. Their Y chromosome haplogroups J2B2A1, J2A1A4B, T1A1B, B2B1A, and E1B1B trace paternal lines to Anatolia, the Levant, and North Africa. On the maternal side, mitochondrial DNA lineages N1B1A1, H, T2C1C, and U1A1 point to a blend of Western Asian and Southern European ancestry. Principal component analysis and QPADM modeling placed these individuals squarely within the cosmopolitan genetic cluster identified in the 2019 Roman-wide study, where nearly 2/3 of imperial-era Romans carried Eastern Mediterranean ancestry. The genetic signals, Anatolian Neolithic, Levantine, and Iranian components, suggest that Pompeii's population was less a closed Italic village and more a bustling port city shaped by centuries of migration, trade, and forced movement. The same DNA markers found in the city's victims echo across the empire, from Alexandria to Carthage to the Aegean coast. This diversity, invisible to the eye and ignored by early excavators, now stands at the heart of Pompeii's story. The people immortalized in plaster were not a static, homogeneous community, but the descendants of travelers, merchants, freed slaves, and migrants. Their genetic mosaic upends the myth of a purely local tragedy and reveals Pompeii as a crossroads of the ancient world, its final moments frozen, but its population anything but fixed. Hidden inside the skull of a man found in the house of the craftsman, a fragment of petrous bone held secrets untouched for nearly 2,000 years. When researchers sequenced his genome in 2022, they discovered not only his ancestry, but also the shadow of a disease, Mycobacterium tuberculosis. The pathogen's DNA appeared among the ancient human reeds, clear evidence of spinal tuberculosis known as Pott's disease. CT scans of his vertebrae revealed the classic collapse and destructive lesions, a silent record of years spent in pain, his back slowly giving way. This was no healthy Roman cut down in his prime. Chronic illness would have limited his movement, perhaps forcing him to stay indoors as the first ash began to fall. The same study identified signs of osteoarthritis in a female victim from a neighboring house, another case of restricted mobility. These findings strip away the myth of robust, untroubled lives. They reveal individuals already marked by frailty, their final moments shaped as much by illness as by the violence of the eruption. In the final hours of Pompeii, bodies were locked in twisted, grasping poses, faces contorted, limbs frozen mid-motion. For generations, these shapes were read as scenes of agony or familial embrace, their drama heightened by the plaster that preserved every muscle and finger. Yet the true cause of these postures lay not in slow suffocation or drawn-out suffering, but in the physics of heat. DNA extracted from the casts carried a silent record of their final moments. Patterns of cytosine to thymine damage matched laboratory models for exposure to 250 to 300 degrees Celsius. Forensic studies confirmed that such temperatures trigger cadaveric spasm, an instant, irreversible contraction of the muscles at the moment of death. The victims died in seconds, their bodies caught mid-action by a thermal shock as sudden as it was final. This evidence does more than clarify how the Pompeians died. It demands a reckoning with the stories told about them. For over a century, museum labels and guidebooks assigned gender, kinship, and emotion based on posture and jewelry, projecting Victorian ideas of family and femininity onto anonymous remains. The DNA results and thermal signatures now make these narratives untenable. Curators face a responsibility to revise displays, update captions, and acknowledge the limits of past interpretation. The casts are not just relics of an ancient disaster, but also of the biases that shape their preservation. Science has made it possible to honor the dead with accuracy, replacing comforting myths with the truth written in bone and ash. Today, DNA evidence leaves no room for comforting illusions. Pompeii's victims are no longer stock characters in Victorian tragedy. They are individuals, descendants of immigrants, revealed by their own genomes. As technology rewrites ancient history, the real mystery is how much of our present we still see through inherited myths. Share your thoughts below.